Thanks for logging. My name is Abby. I am a PhD candidate in the Miller Lab here at the University of Michigan. And my project focuses mostly on mitochondrial and peroxisomal beta oxidation in the RPE and how these two organelles work together to accomplish really essential lipid metabolism in the RPE. I want to first thank Constantine from Matt Benson's lab up in Canada for giving such a wonderful presentation last time we were together. I was secretly hoping that he would give a great overview of peroxisomal biogenesis and beta oxidation, and he really nailed it. So thanks, Constantine, for the warm up, and that will only help everyone understand some of the peroxisomal beta oxidation that I'm going to talk about today. Our lab is first and foremost an age-related macular degeneration lab, so I'm going to start there. Several of us are familiar with AMD, which primarily affects the RPE, the retinal pigment epithelium. We're all familiar with this kind of diagram. Under normal circumstances, the RPE receives a lot of nutrients and oxygen from the choroid at its basal surface and secretes waste products back into the choroid. It also transfers nutrients, especially glucose, up to photoreceptors, and it phagocytoses photoreceptor outer segments, which are full of lipids. It also transfers back the lipids up to the photoreceptors, especially DHA. So it's this very complex, metabolically active tissue in the retina. But over the course of AMD, the RPE develops extracellular lipid-rich deposits called drusen below the basal surface, and these deposits have a lot of proteins and lipids in them that kind of act as a barrier between the RPE and the choroid, so that the tissue gets less nutrients, less oxygen, and this can progress as far as what we call geographic atrophy, in which whole chunks of the RPE start to die, which leaves photoreceptors without the nutrients they need as well. So photoreceptors will also die. And this leads to a loss of vision with a pattern that looks something like this. You can see AMD vision on the right side here. Change my zoom view. On the right side, you can see AMD vision from a patient, and on the left, you can see what a normal person would see. Because this is happening in the macula, which gives you your most clear, crisp vision, patients also have pretty blurry view around that inner blind spot. So this also affects about 50% of Americans over the age of 80 to 85. And we're really interested in looking into the lipid metabolism and other metabolic processes that are happening in the retina that actually go wrong and contribute to AMD, to hopefully find a new avenue of treatment for AMD in the future. The RPE is a really special tissue. It's a really unique epithelium. And one of the ways it's really unique is that it receives a lot of lipids from both the basal surface and the apical surface throughout the day. So starting with the apical surface, of course, there are photoreceptors that sit between the microvilli of the RPE. And the outer segments of these photoreceptors look something like this under an electron micrograph. These outer segments are just stacks and stacks and stacks of these membrane discs. And these membrane discs, of course, are full of a lot of phospholipids that specifically have a lot of very long chain polyunsaturated fatty acids. And on the basal surface, the RPE is highly enriched in lipoprotein particle receptors. We're all familiar with lipoprotein particles that look something like this. And the fatty acids within these lipids are typically long-chain fatty acids that are saturated or monounsaturated. So these are the palmitate, stearate, and oleate that we most often think of when we think of fatty acids. And I mentioned these specific fatty acids that the RPE is receiving all the time to show that not only is the RPE receiving a high volume of lipids every day, but also a really wide variety of fatty acids that it has to be able to break down and beta oxidize to maintain its health and to maintain the health of the photoreceptors that it supplies metabolically. So to review really quickly, fatty acids are typically come in even number carbons in their hydrocarbon chain, and we categorize them based on the length of those hydrocarbon chains for the most part. So short chain, medium chain, and long chain fatty acids are pretty common. Again, long chain fatty acids include those palmitate, oleate, and stearate that we commonly think of. However, there are some other not as common fatty acids, including very long chain fatty acids, like those found in photoreceptor outer segments, dicarboxylic acids that have a carboxyl group on both ends of the fatty acid, as well as this very strange odd number 15 carbon branch chain fatty acid called pristanic acid. And this is not found in abundantly in the RPE. However, it will be a useful experimental tool that I'll talk about in a little bit. So the RPE is receiving long chains from lipoprotein particles and some in the photoreceptor outer segments, and very long chain polyunsaturated fatty acids from outer segments. And I'm going to divide these fatty acids into two groups. Short, medium, and long chains are targeted to the mitochondria for beta oxidation, while very long chain, dicarboxylic and pristanic acid, and a few other fatty acids are targeted to peroxisomes for beta oxidation. And typically, based on the literature, we think of these two pools of substrates as completely separate. The mitochondria takes care of these fatty acids and only these fatty acids, and peroxisomes take care of these fatty acids and only these fatty acids. However, there is emerging evidence in the literature, including some data that I'll show you today, that these two pools aren't entirely distinct, that they overlap, and specifically that peroxisomes can actually beta oxidize some of the fatty acid substrates that are typically targeted to the mitochondria. So the relationship between these two organelles is really important, and they actually do work together to completely beta oxidize all the fatty acids that the RPE is receiving. However, during AMD, the RPE mitochondria become highly dysfunctional, and we have a lot of evidence for that in the literature to show exactly how they become dysfunctional, which includes a lot of mitochondrial DNA damage, increased ROS production, a decrease in size, as well as a decrease in number of mitochondria in the RPE, and fewer cristae and membrane ruptures within the mitochondria, both as we age and a little bit faster as AMD progresses. And we've seen this with plenty of evidence in the literature, including electron micrographs of AMD patients, RPE and retina. But the one mitochondrial function that I've been talking about and that you don't see on this list is beta oxidation. And we asked ourselves, or rather, one of the first questions that I asked Jason after I joined the lab is, we're talking about all these other ways that the mitochondria is becoming dysfunctional, but what happens to beta oxidation? And do we know if beta oxidation in the mitochondria is specifically dysfunctional or damaged? And the reality is we don't know. We don't have enough evidence in the literature to show whether or not it's beta oxidation specifically that is dysfunctional during AMD. So instead of going down that rabbit hole and trying to investigate ourselves, we've instead turned to patient data to try to figure out, does the dysfunction of beta oxidation specifically contribute to retinopathy, or more specifically, RPE atrophy? So if we look at different patients with single enzyme mutations in mitochondrial beta oxidation, we get this list with different beta oxidation steps within the mitochondria. And these genes are associated with each of these steps in mitochondrial beta oxidation. And on the right, you see here, each dash says, no, there is no retinal phenotype as in patients that have mutations in these genes do not present with a loss of vision or retinal degeneration. There is, however, one single mitochondrial beta-oxidation disorder that does result in a retinal phenotype, and this is specifically RPE atrophy. It's a mutation in the HADHA gene involved in hydration and dehydrogenation step. However, we've heard a little bit about this mutation specifically that leads to LCHAD deficiency in this seminar series from Dr. Melanie Gillingham at OHSU. But she's pretty confident that it's not the loss of beta oxidation specifically that leads to RPE atrophy, but rather a buildup of fatty acid intermediates that are highly oxidizable and end up disrupting membranes and increasing ROS production that actually leads to RPE atrophy. So based on all this patient data from specific single enzyme mutations in mitochondrial beta oxidation, we're hesitantly concluding that it's not mitochondrial beta oxidation or lack thereof that leads to RPE atrophy, but rather all of the other dysfunctions that happen in the mitochondria over the course of AMD that contributes to the progression of AMD. But that changes a lot when we look at peroxisomal beta oxidation. If we look at the same steps of beta oxidation that the peroxisome does, the peroxisome actually uses a different set of genes and enzymes to perform its beta oxidation, but it is the same few steps. So if we look at these steps in the peroxisome, what we see is that a majority of them lead to a retinal phenotype or retinal degeneration. And this is the same is true of peroxisome biogenesis disorders like those that the Benson lab studies. So mutations in PEX genes that contribute to peroxisome biogenesis also lead to a loss of vision and retinal degeneration. So based on these other patients that don't have AMD but do have specific genetic mutations associated with the peroxisome and the mitochondria, we can conclude that it's actually peroxisomal beta oxidation that is really, really crucial to RPE integrity, and not necessarily mitochondrial beta oxidation, but other dysfunctions within the mitochondria. So
Hey, Abby,
yeah,
sorry to interrupt, but for the mitochondrial genes you listed there,
yeah.
do they have other human phenotypes when they're mutated?
Oh, definitely. These mutations can definitely be really harmful to the rest of the body, but none of them except this one that says yes have a loss of vision.
Do the patients like live very long when they have recessive mutations in these genes?
Some of them do. I'd have to look into it more about how long they live, and I think there's probably a bit of a variety and different severities within these different mutations. These ones specifically, the LCHAD deficiency or HADHA mutation, they can live into adulthood, but they often have sudden cardiac arrest and die as young adults, unfortunately. So I think there's a bit of a range amongst these mutations.
I'd just be careful to dismiss like the importance of mitochondria for retinal health, or at
Yeah.
least RPE, because you don't see retinal phenotype. If
Mhm.
possible, the patients are dying prior to being able to complain of bad vision, or if
Yeah.
they're not assessing their vision because they are dying at like 8 months of age or something.
No, that's a great point. We should look more into the amount of their timeline, their lifespan. Totally. That's a great point, Eric. Thank you.
So just to clarify, Eric, the point being made here is not that mitochondria aren't important for the RP. They're definitely important for the RP. Tons of mitochondrial DNA disorders cause RPE degeneration, almost as a primary tissue affected. It's the beta oxidation disorders of the mitochondria that don't necessarily lead to a retinal phenotype. So that we're not dismissing mitochondrial importance. We're just asking how important is the process of mitochondrial beta oxidation to the everyday sort of health of the RPE.
I understand. It's just like I looked up CPT1A real quick on OMIM, And those patients were like die in childhood, very early, like within a year. So it's not it's hard to determine if a kid that young has a retinal phenotype, right? Because they have so many other systemic issues. Who knows if you're not seeing a retinal phenotype just because they die so young? This is what we ran into with like the GLS knockout.
Yeah.
Is there's very, very few humans who survive when GLS is completely lost.
I
So
I
yeah.
agree with that point overall for sure. Melanie is on the line and she can comment this, which we should have her since she's the literally a world's expert at mitochondrial beta oxidation disorders. But there's definitely a range of survival, including many that live quite long but have to avoid starvation. But Melanie, do you want to comment a little bit on the range of lifespan for beta oxidation disorder?
Sure, sure. CPT2 patients live well into adulthood. They're 60, 70-year-olds with CPT2. VLCAD also, we have, 40, 50-year-olds, but the genes were generally described in the 90s. So most of the older age patients are 40, 50 years old that have survived. MCAD also, as far as we know, lives a normal lifespan. I would say the ones that are the most fatal are the CPT1A severe forms, CACT, and the severe forms of a lot of them will die younger. And then LCHAD is a little bit on the more extreme side. And many of those patients also struggle with cardiac symptoms in their early adulthood.
And to just maybe, I think Eric's point is very well taken, but just to add a parallel on the peroxisomal side, there's just equal severity to many peroxisomal,
Very,
disorders in which they die quite
very
young,
young.
and yet the phenotype manifests even in the retina, from
Yes.
often from an early age. So it's definitely not quite, it's not an apples to apples comparison, but just across all the disorders and the fact that many mitochondrial beta oxidation disorder patients can live a long time, and some peroxisomal beta oxidation disorder patients can live longer. Sort of all comers, you're getting much more retinal phenotype in a peroxisomal beta oxidation disorder set of patients than in a mitochondrial beta oxidation disorder.
Yes. Yeah, I would agree.
And do you also, my general understanding is that you can have brain phenotypes when you really don't have retinal phenotypes in a lot of these, not just necessarily the beta oxidation disorders, but in the mitochondrial disorders, which is really quite interesting that brain will be affected, but not retina.
Yeah, that's interesting. I can't speak necessarily to the mitochondrial beta oxidation disorders, but peroxisome biogenesis and beta oxidation disorders often come with neural phenotypes. Yes.
I would say the fatty acid oxidation disorders do not.
Yeah.
So their brains are typically not affected.
But Louis, I agree with you. There are definitely mid and MELAS, that have no that don't necessarily have any retinal degeneration, but have, of course, profound,
Right.
neuronal. I think that's probably what you're referring to. Yeah.
Right. Yeah, exactly.
Absolutely. Yes, thank you guys, and thank you, Eric, for reminding us about that. That's a great point. Again, these two organelles have to work closely together in all tissues, but especially in the RPE, to be able to beta oxidize all the different fatty acids that the RPE is receiving every day. And that system looks something like this. Starting with the peroxisome, it receives its substrates, such as very long chain fatty acids, and it imports them with an ATP-dependent transporter that's much more simple than the really complex system that mitochondria use to import fatty acids. They perform beta oxidation with their own set of enzymes, but rather than completing beta oxidation or getting all the way down to only acetyl-CoA, they actually bite off acetyl-CoA chunks until they get down to a medium chain fatty acid of about 8 to 14 carbons. And they say, okay, I'm done, get out. They kick out the acetyl-CoA and the medium chain fatty acid, which has to go back to the mitochondria for complete beta oxidation. The mitochondria receives substrates both from the peroxisome as peroxisomal beta oxidation products, as well as their own substrates that are directly targeted to the mitochondria, like the long chain fatty acids of the lipoprotein particles that the RPE receives. Mitochondria breaks down using their own beta oxidation machinery that we're more familiar with. All of these substrates, all the way down to acetyl-CoA, which is the final product. It's a 2-carbon fatty acid that we're familiar with. Acetyl-CoA, of course, fuels the TCA cycle, which fuels the electron transport chain that consumes oxygen while it produces ATP. And the one part of this diagram that's missing is that peroxisomes actually also consume a little bit of oxygen. In the first step of peroxisomal beta oxidation, rather than exchanging NAD plus and NADH, they actually consume half of an oxygen to produce hydrogen peroxide that it has to then neutralize. So we have some oxygen consumption from both of these organelles that we're going to take advantage of in the main experimental technique that I'll talk about here in a little bit. However, during AMD, mitochondria are severely dysfunctional, and whether that affects beta oxidation specifically or not, the mitochondria certainly are not as efficient as at breaking down their substrates. So what happens to the long-chain fatty acid substrates specifically that are coming especially from those lipoprotein particles? Which leads me to the big question in my project, which is, can peroxisomes compensate for the mitochondrial dysfunction that's occurring in AMD in the RPE? And that's a pretty big general question that may take a lifetime to answer. So I've only been in the lab for about a year and a half. So instead, I'm going to ask a bit of a smaller, easier to tackle question. Can peroxisomes beta oxidize long chain fatty acids specifically? These fatty acids are directed to the mitochondria normally, but can peroxisomes beta oxidize them for the mitochondria instead? And in order to answer this question, we have to be able to detect peroxisomal beta oxidation and differentiate it from mitochondrial beta oxidation. And the established method of doing that is called acylcarnitine profiling. Acylcarnitine is just a fatty acid with a carnitine moiety attached. And this is performed with mass spec that is often for us performed by Dan himself. So thank you, Dan, for putting up with our requests. And to do acylcarnitine profiling, we take advantage of the production of medium chain fatty acids from the peroxisome because the peroxisome often tags its products with a carnitine moiety. So we're able to detect these if we block the entry to the mitochondria. But first, let's start with the results we get in our negative control when we have no intervention and no inhibition of the mitochondria. We simply feed cells a 13C tag palmitate that is targeted to the mitochondria. It runs through beta oxidation to produce acetyl-CoA. And the results we get in our negative control look like this. There's a large pool of C16, which is our substrate, our palmitate that we fed the cells, and a large pool of C2, which is essentially acetyl-CoA that the mitochondria is producing through its own beta oxidation. There's no buildup of medium chain fatty acids because beta oxidation is allowed to run straight through. There's nothing blocking the mitochondria or its beta oxidation. So all we see is the substrate that the cells have not finished using up after 4 hours, and the product of beta oxidation, acetyl-CoA. But to take advantage of the medium chain fatty acid products from the peroxisome, what we're going to do is use an inhibitor called LAC or L-aminocarnitine. And this is a CPT2 inhibitor. So it blocks both the long chain fatty acid substrates like palmitate from entering the mitochondria, and it also blocks the medium chain products from the peroxisome because those have a carnitine moiety on them. So if you block CPT2, that removes the carnitine moiety, it can't enter the mitochondria, or it can't complete beta oxidation because it can't be exchanged for a CoA moiety. So we feed cells 13C tag palmitate along with our inhibitor LAC. We wait for 4 hours, collect the lysates, and send them off for mass spec. And the results we get look like this. Again, there's a big pool of C16 or palmitate that we fed the cells. And it is a little bit larger than the negative control DMSO. There's also a smaller pool of acetyl-CoA buildup because the mitochondria is blocked, so we don't get as much acetyl-CoA. And there's a buildup of these medium chains, 10, 12, and 14. And that indicates to us that the peroxisome was able to beta oxidize some of the palmitate, and it produced medium chain fatty acids that were not able to enter the mitochondria. And this replicates almost exactly what a few others have shown in their own data, especially with CPT2 knockout cells. Specifically, one of
Abby?
our, yeah?
This is Dorota. I'm super interested. Very nice work. But just question: what cells are you using?
Yes,
exactly.
these are human primary RPE cells.
Thank you.
Yeah. So one of our collaborators, Sandra Hooten, over at Mount Sinai in New York, has done very similar acylcarnitine profiling with his own CPT2 knockout cells that were not RPE cells. So it was really encouraging for us to see that we could replicate those results in our own cell type, and with this 13C tagged palmitate. So we've confirmed that we can do the established method, acylcarnitine profiling, and we will be doing more of this in the future because it does more concretely point toward peroxisomal beta oxidation. However, one of the drawbacks is that this is just a single time point snapshot of that beta oxidation. So we're not getting a very holistic view of how the cells process the fatty acids that we're giving them. And if you've ever cultured RPE cells, you'll know that they're very difficult to grow up. They're difficult to differentiate from iPSCs. And because they're post-mitotic, we keep them around for years at a time, changing their media twice a week, and just leaving them in their plates. We don't replate them at all. So they're very precious cells that we do our best not to sacrifice or lyse whenever we can help it. So we are very picky about when we perform acylcarnitine profiling, so that we're not wasting any cells. And in order to get a more holistic view of the lipid metabolism we're trying to measure and to avoid lysing our cells, the method that our lab has turned to is long-term oxygen consumption rate, or OCAR. Many of you have probably heard of a seahorse that measures OCAR over a period of minutes to hours. But this crazy device here that we use actually measures OCAR over periods of days to weeks if we keep changing the media often enough to keep the cells alive. So this crazy device is called a Recipher from Lucid Labs that we are fortunate enough to have 6 of in our lab. It involves this 3-part sandwich that includes a cell plate, a 96 well plate with cells plated in a very specific configuration. And the sensing lid that sits on top of the cells has these oxygen probes that measure oxygen throughout the media column above the cells. Using the device that sits above the sensing lid, it has these automated screws that control the height of the sensing lid to measure oxygen at the top and bottom of the media column. And that the gradient of oxygen throughout the media column is how it calculates OCAR. So if there's a really severe gradient, as in really low oxygen at the bottom of the media close to the cells and really high oxygen at the top of the column where atmospheric oxygen is available, then OCAR is also really high because cells are consuming a lot of oxygen. So we're able to detect how much oxygen cells are consuming, which is a measurement of both mitochondrial oxygen consumption as well as some peroxisomal oxygen consumption. And the results that we see from this device look something like this. We always start with palmitate, which looks like this. And we use palmitate as a positive control because it's the most abundant fatty acid in the body and in those lipoprotein particles that the RPE receives at its basal surface. Our negative control in gray here is just the same serum-free media that we use for palmitate, but with no fatty acid added. And the 2 parts of these graphs that we're going to be looking at are highlighted here in gray, one at the start of the run and one closer to 25 hours at the end of the run. And these 2 parts of the graph first indicate that a bolus of mitochondrially targeted fatty acids like palmitate here in blue trigger a really steep sudden rise in OCAR. And that's because without any inhibitors, palmitate is allowed to enter the mitochondria and run through beta oxidation really quickly to produce acetyl-CoA that feeds the TCA cycle that fuels the electron transport chain that consumes oxygen. And that happens really quickly when there's this 300 micromolar bolus of palmitate to the cells. They first reach for palmitate, OCAR spikes really suddenly. At this later time point, closer to 25 hours, palmitate has run out and cells start to reach for glucose, and OCAR decreases because whether glucose is aerobic or anaerobic, it's not going to consume as much oxygen as that sudden beta oxidation event did. So OCAR starts to decrease, and we've shown that glycolysis increases around this time point as well with another device we have called a lysel mode that senses the glucose and lactate concentration in the media. And we know that glycolysis increases or glucose consumption increases at this time point when OCAR is decreasing because all the palmitate has been used up.
Can I just ask, what concentration of glucose are you using when you give the palmitate bolus?
Yes, we have a 5 millimolar glucose
5
in our
millimolar,
serum-free media. Yeah. The cells also have other mitochondrial substrates like amino acids in the media as well. So
Okay.
they can actually survive with even the control that didn't have this palmitate bolus. They survive, and this control will keep climbing up a little bit above the palmitate typically for quite a while because it hasn't been relying on the palmitate bolus. So it had to turn to glucose and other mitochondrial substrates from the beginning, which is why it's slowly but steadily increasing throughout this part of the run.
Yeah, it's just really interesting that with sufficient glucose, it's then using palmitate. So it's preferring palmitate
Yeah,
over the glucose.
no, it's very interesting. So this is
I just will add, because this preceded Abby joining the lab, so she would unfortunately didn't know this stat or Pearl, but to Lois's point, we've done the labeling with glucose and palmitate, identical conditions, but the glucose was on, or the label was on glucose, or the label was on palmitate. And you're correct that on the initial sort of feed and bolus, the labeling into TCA cycle intermediates comes first or most dominantly from the palmitate and later from the glucose. If you have both around, yeah.
Yeah, can I comment on that too? So I'm sure you all remember the work from Jean Haidu when he saw that there is immediate usage of glucose and there is almost no glucose later on in the media, right? So I just wonder if we shouldn't consider not having glucose in those for our RP culture, just bringing it to maybe later discussion. For having more conditions that are in the eye where glucose is all like just shuffled to the photoreceptors, just something to consider. But since we have this nice discrimination of usage, I think we can discriminate what's going on. Thank you.
Yeah, no worries. So this trend from palmitate, which is targeted to the mitochondria, is a pretty consistent trend. Palmitate is actually a little bit unique in that it always causes the largest spike out of any of the fatty acids that we've seen at this early time point. But if we look at other mitochondrially targeted fatty acids, such as stearate and oleate, we're going to see a pretty similar trend as well. Stearate, of course, is the saturated long chain fatty acid that's also pretty common in lipoprotein particles, as well as oleate that's monounsaturated. And when we run these on Recipher, what we see is this. They look really similar to palmitate with this spike in this early time point that later drops to meet the control closer to 25 hours. So this really consistent trend points toward mitochondrial beta oxidation to us. And I say that specifically because the peroxisomally targeted fatty acids that we run on Recipher look a little bit different. So in contrast to this pretty consistent trend, when we look at peroxisomally targeted fatty acids that we utilize to investigate peroxisomal beta oxidation, they're going to look a little bit different. starting with DHA, that is really important to retinal health. DHA in vivo RPE are designed to preserve DHA. They repackage it from the outer segment phagocytosis into triglycerides and send it back up to photoreceptors. But in vitro, we've seen that it does actually raise OCAR, which indicates that it's being beta-oxidized. And pristanic acid is our really weird 15-carbon branch chain fatty acid that's not necessarily abundant in the RPE, but we know it's targeted to the peroxisome, so we also use it to investigate peroxisomal beta-oxidation. And when we run these compared to palmitate on Recipher, the results look like this. So at this early time point, the peroxisomally targeted fatty acid, both of these, are really low, much closer to the control. Their OCAR rises much more slowly, but at this mid time point, about 10 to 15 hours, it crosses above palmitate and plateaus at this higher OCAR for a long period of time, much longer than this 25 hours that I have here. And this looks pretty much the same in pristanic acid as well. So now we're seeing this distinct difference between the mitochondrially targeted and peroxisomally targeted fatty acids and how they're processed in the cells over this period of time. If we look at two more peroxisomally targeted.
Well, can I ask a quick question? Do you know the time courses with which the palmitate and the DHA and the pristanic acid get depleted from the media in these experiments?
What we've seen, the data we have so far will show approximately when the cells reach starvation and all of the substrates have been utilized, which in my experience is about 70 to 80 hours after treatment. We have some acylcarnitine profiling from palmitate specifically, and at 16 hours at this time point right here, there is still a little bit of palmitate left over. It hasn't necessarily all run out yet. But we don't have enough tracing studies, I think, to say exactly when it's entirely run out.
But can you just measure it in the media and see how?
We can measure in the media, and we have started doing some simple fatty acid plate assays to see if there's generally any fatty acids in the media, although we haven't gotten far enough to show exactly when it's all completely run out, I think. But our postdoc John has started doing some of those plate assays.
I was just wondering if they're all depleted at about the same rates, or if they're used different rates. Yeah.
That's a great question. I would guess that the peroxisomally targeted fatty acids probably run out slower because we're theorizing that this slower rise in OCAR is because it just takes longer for these fatty acids to go to the peroxisome, make it all the way through, and go back to the mitochondria. But I think we don't have quite enough data to fully describe that yet, so I won't make any big.
Can I just ask, have you looked at breakdown products of DHA? I'm only asking because is it not possible that DHA is actually stabilizing membranes to increase OCR rather than being used as a fuel?
That's certainly possible. This pattern is so consistent with other peroxisomally targeted fatty acids, though, that we so far have concluded that it is raising OCAR, and this is pretty new data, so I'd say that we haven't talked it through all the way yet. I kind of surprised Jason putting this in here. But that's certainly something that we should look into, and we're planning to do more DHA tracing studies in our cells in the future. Yeah.
Oh, it's very interesting data.
Mm-hmm.
I think Dr. Hurley's point was very important because we have observed N=1 result that DHA, when you add DHA the first 48 hours, it spare a little bit of glucose consumption. But after 48 hours, if you wash it away, there's a very strong spare of glucose after you just replace with new media. So I think the time point is important. Yeah, that's one comment.
Yeah, thanks, Richard.
Okay, I just have a comment that just completely a little bit different because we also see DHA connected to the acylcarnitine, I mean, in the form of acylcarnitines. And I wonder how, I mean, whether you tried these experiments with the inhibition of the mitochondrial oxidation.
Yes, I have performed this with etomoxir, actually a CPT1 inhibitor. And these lines, at least in these early time points, whether DHA is inhibited or not, those lines are right on top of each other. So that's a nice check, and I'll show some similar data here in a minute to show that when we inhibit the mitochondria, most often the peroxisomally targeted fatty acids are not affected at all, and their rate of OCAR remains the same.
Thank you.
Yeah.
Abby,
Mhm.
Martin Paul here.
Yeah.
To follow up on Dr. Smith's earlier question with regards to the, Dr. Hurley, with regards to substrate depletion at 300 micromolar, are we sure that really the substrate is completely depleted from media and cells?
I don't know that we can be entirely sure yet because we haven't done as much acylcarnitine profiling and other tracking as we're hoping to do in the future. So hopefully we can get closer to answering that question. But we have, I think in the past, others have done a lot lower concentration of DHA feeding that doesn't necessarily raise OCAR. So Jason and I were just talking about this this week. Is it also somewhat concentration dependent, and do our in vitro cells preserve DHA until there's so much of it that they're pushed to beta oxidize it so that it doesn't build up? But yeah, I don't know that we have enough data quite yet to say if any of it is spared and when it runs out in this process.
All right. I mean, yeah, your thought is what I'm thinking of as well, that probably the high bolus kind of triggers the cell to stabilize the system, so they end up metabolizing the fatty acids first before, because in my experience, you hardly deplete the fatty acids from the media. And at 300 micromolar in 72 to 80 hours, I will be surprised that you have complete depletion in the media and the cells.
And I just to step in, because again, this is it's not fair to Abby because it's preceded her joining the lab. But just to one question that was asked was, are fatty acids depleted, and at what rate are they depleted? And I think that's a common theme among the questions. And the answer is that palmitate is the actual palmitate, when we add 300 micromolar palmitate, is on average depleted somewhere between 12 and 20 hours from the media. Now that doesn't mean that it's depleted from the cell system itself, but it's definitely depleted from the media. And some initial data suggests that depletion rates correspond well to where the spike in OCAR is. So, palmitate's spike in OCAR is earlier, and the palmitate's being sort of avidly consumed and depleted, and it's contributing to this high OCAR initially. It is a very good point, and on our to-do list to do the exact same thing with the peroxisomally targeted fatty acids to understand what their depletion rate is. And one of the things that's on John, the postdoc's list, is to basically figure out whether these simple fatty acid enzymatic kits, which allow you to do stuff very quickly, can work with the very long chain fatty acids. If they do, it's just an easy enzymatic assay to check for their depletion rates and how they correspond to this delayed rise in OCAR. So we definitely, and then of course, I think to Lois's point, we are 100% sure that we need to do a lot of labeling and tracing experiments to complement any results that we see here in this early OCAR stuff, to be sure that what we see with OCAR really corresponds to the degradation of these fatty acids.
Yeah, and mentioning the labeled peroxisomally targeted fatty acids, John and I are in cahoots to push Jason to get tagged DHA, as well as this very long chain saturated fatty acid C26 that we've talked a little bit about. So in our lab, but if we look at other peroxisomally targeted fatty acids, DHA and pristanic acid, maybe DHA can be considered very long chain, although it's a little bit on the cusp between long and very long. So we've also looked at other very long chain fatty acids. First, C34,5, which has 34 carbons and 5 double bonds. It's a monster of a fatty acid, and I've heard that Martin Pall likes to call it DHA on a stick, because it has this DHA shape at the end here, but because it has so many more carbons, it's got this little lollipop kind of shape. And we chose this very long chain PUFA because it's pretty abundant in photoreceptor outer segments. We also chose this C26 saturated very long chain fatty acid because it's pretty abundant or elevated in the plasma of peroxisome biogenesis disorder patients. So well, if we look at Recipher results from both of these, we also see a similar trend of the peroxisomally targeted fatty acid has this slower rise in OCAR that crosses above palmitate and extends at this plateaued level. And if we looked at a much longer time point, this C34,5 actually will stay above palmitate for a really long time because it has so much more carbon in it that it can feed first through the peroxisome and then to the mitochondria from those medium chain products. And when we look at C26, this is hot off the press data from last week that our postdoc John got, that he said that I could use today. It's very preliminary, but it's super exciting for us because we've been working on getting C26 into solution and conjugated to BSA like we do for all of these other fatty acids. And he finally got it to work after working on it for months and months and months. So this is fresh preliminary data from John. Again, C26 causes this slower rise in OCAR, but it catches up to palmitate and crosses above palmitate. Sometimes we see these funny little dips in our OCAR trends. I think these particular dips were my fault last week. When we opened the door to our incubator, we disrupt the oxygen and CO2 levels, and you get this little glitch in the Recipher data. So those little dips are probably my fault when I opened the incubator. But this is really exciting for us that we were able to get it into solution and conjugated, and we'll be using this particular fatty acid more in the future. And maybe if we can get it labeled, then we can do some more fun tracing studies as well.
Oh, and Abby,
Mhm.
sorry, it's just Matt Benson here. Yeah, this is super cool. Do you mind if I just ask a quick question about
Yeah, go
this?
for it.
Because the time course is quite delayed for the C26. Like, do you suspect it just takes up a longer time for it to be internalized into the cell, or is that something that you'd anticipated?
It's not necessarily something that we anticipated because the C34,5 does kind of follow the same time course that we've seen in other ones. So I'd say that we haven't run the C26 quite enough times yet to see if this is actually a real trend that we're seeing, because the control line is also a lot more delayed than we typically see. So honestly, I would guess for now that because this is very preliminary and we are just now working on our protocol to get this into solution and conjugated, that this could be a little bit of a fluke in the time period of when C26 gets metabolized in relation to palmitate. If it is metabolized later than C34,5, it could be a delay in reaching getting into the cell and getting to the peroxisome. And we've actually been talking about some digitonin-based studies that we might do in the future to investigate if peroxisomally targeted fatty acids, especially these greasy, very long chain saturated fatty acids have trouble just getting into the cell, even though they're conjugated to BSA. So we might be addressing that in the future with some digitonin membrane disruption assays to see if that equalizes the time period or not. I hope that
Okay,
answers that
thanks.
question, Matt. Thank you. So we have all this data to show that there's a big difference between the Recipher, these OCARB curves between mitochondrially targeted and peroxisomally targeted fatty acids, and they're pretty consistent trends. So we've been using these to differentiate between these two organelles and their beta oxidation processes. Our next question is, can we direct long chain fatty acids that normally go to the mitochondria to the peroxisome? And we've done this with our acylcarnitine profiling using a CPT2 inhibitor LAC. In this case, we're actually going to use a CPT1 inhibitor, atomoxir. And because we're inhibiting CPT1, what that does is block the entry of long chain fatty acid substrates into the mitochondria. Hopefully, it redirects them to the peroxisome because they have to be broken down somewhere. And then the medium chain products from the peroxisome actually already have a carnitine moiety on them. So because CPT1 is in charge of putting on a carnitine moiety or exchanging it for CoA, this medium chain fatty acid product that already has carnitine on it is able to bypass CPT1 that's being blocked by atomoxir and enter the mitochondria. Hopefully, that's the theory. So when we apply palmitate or other mitochondrially targeted fatty acids along with atomoxir, the patterns we get look like this. At this early time point, inhibited palmitate has a much lower OCAR. There's this huge gap between our control uninhibited palmitate and palmitate with atomoxir. At this pretty consistent 10 to 15 hour time point, the atomoxir inhibited palmitate crosses above our control and plateaus at a higher OCAR for quite a while. Later on in the time point, they both will drop in OCAR around the same time because there is the same amount of carbon that we're feeding the cells. It's just that we're altering the OCAR kinetics when the palmitate and where the palmitate is being beta oxidized. The same is true of oleate. When we inhibit with atomoxir, OCAR is much lower at this early time point than our uninhibited oleate, and later crosses above the uninhibited form and plateaus at a higher OCAR. So this is really exciting, and these OCAR curves look a lot like the peroxisomally targeted fatty acid curves that we've looked at before, which is part of what indicates to us that we're seeing that these long chain fatty acids are being beta oxidized by the peroxisome. And we'll be confirming that more thoroughly with acylcarnitine profiling in the future. But because Recipher is pretty easy for me to run in lab at the drop of a hat, and acylcarnitine profiling is a little more complicated, I've been running this device almost every week since Christmas last year. So we have quite a bit of data to contribute to this project from the OCAR standpoint. So as a double check, when we apply atomoxir to peroxisomally targeted fatty acids like pristanic acid, we see that these two lines are right on top of each other. They track together because pristanic acid and other fatty acids like the very long chains and DHA are not sensitive to atomoxir, meaning that atomoxir is effective at blocking those long chains at the mitochondria, and pristanic acid is not going to the mitochondria at all, so it's not sensitive to atomoxir because it doesn't need to use CPT1. It's not going to the mitochondria. So that's a nice double check. And our working model currently is that during mitochondrial inhibition, long-chain fatty acids are redirected to peroxisomes for beta oxidation. And the reason we're concluding that is because of preliminary acylcarnitine profiling data, as well as the similarity between these curves. This dotted blue line is palmitate with atomoxir inhibition. and the orange is pristanic acid that we know is going to the peroxisome. So,
Sorry, can I ask a quick question?
yeah.
So how do you distinguish? So I totally buy that the shape of sort of both the orange curve and the dotted blue line are indicative of slowed fatty acid oxidation. And how do you distinguish between slowed mitochondrial fatty acid oxidation, including something like CPT1 independent, kinetically slow, but CPT1 independent mitochondrial uptake versus, it is going on in the peroxisome specifically?
That's a fantastic question. That's definitely one of the biggest questions that we're going to get from reviewers when we start publishing a lot of this data is how do we know that we're not just seeing altered or slowed down or incomplete mitochondrial beta oxidation. And we have a few things so far to indicate to us that it is peroxisomal, including the acyl carnitine profiling that has been shown in the literature to indicate peroxisomal beta oxidation specifically. We've also titrated etomoxir and LAC, our CPT2 inhibitor, to show that when we increase etomoxir over the concentration I use here, 4 micromolar, it doesn't further inhibit beta oxidation in the mitochondria. However, we're also looking into an iPSC-derived RPE cell line that has a genetic knockout of a peroxisomal beta oxidation enzyme that will be a really useful negative control in the future so that if we run a peroxisomally targeted fatty acid on that cell line, we won't see any rise in OCAR. So that's an ongoing effort in our lab. Jitao is actually working hard on developing those or deriving those iPSCs. So we're trying to take a few steps to really thoroughly demonstrate that we're detecting peroxisomal beta oxidation and not just weird mitochondrial beta oxidation. Yeah, great question, Dan. Thank you.
Can you and maybe I missed it, but can you rule out that that the palmitate isn't getting first elongated and then entering?
Great question. So we are interested in other products outside of, or other oxidation processes outside of beta oxidation, like elongation and even omega oxidation, which is the process of turning a regular monocarboxylic acid into a dicarboxylic acid. And those dicarboxylic acids are directed to the peroxisome. We haven't thoroughly looked into that yet. There is elongation machinery in the RPE. It's not as extensive as photoreceptors that are constantly elongating fatty acids to put into the outer segments in those phospholipids. But that's a really interesting question. We haven't looked specifically, as far as I know, into elongation enzymes. So yeah, that's a really interesting point. I will say now that I'm thinking about it, we sometimes look at the area under these OCAR curves. And for instance, the distance between this pristanic acid and inhibited palmitate could be chalked up to the idea that pristanic acid may only be 15 carbons in the chain, but it actually has 19 carbons because of those branches. So the more carbon we feed, theoretically, the higher the curve of the OCAR, whether that's at this early spike or later on, the more area under the curve indicates more carbon and correlates with that higher carbon, which is why when we run C34-5 that has so much more carbon than palmitate, its OCAR will extend out a lot longer than palmitate. So that also indicates that if palmitate were being elongated, when it's blocked from the mitochondria, its OCAR may rise a little bit later, or it would just be extended a lot longer than this uninhibited palmitate. And we don't see that. These two lines actually will drop pretty close to the same time. Yeah, but that's a great thought. We should look into that a little bit. So to get back a little closer to AMD and some more hopefully future translational projects that we'll have, We're wondering how we can target peroxisomes in the RPE to slow the progression of AMD. Pretty much all the drugs that are on the market, especially for dry AMD, which is what we study, are not very effective at treating AMD currently. So we're thinking about how we can use this altered metabolism and the metabolic activity of the RPE, especially peroxisomes, to treat AMD in the future. And some of the ideas that we've had so far, this list will certainly get longer in the future, include PPAR alpha agonists. However, others in the country have thought of this definitely to treat other retinopathies like diabetic retinopathy. So this is very interesting, but not necessarily the top of our list. If it ends up working really well for diabetic retinopathy, maybe we'll look more into it later. What we're more interested in right now is dietary supplementation of dicarboxylic acids, as well as the upregulation of endogenous dicarboxylic acid production, so that omega oxidation that happens in the ER that turns a regular fatty acid into a dicarboxylic acid without changing the chain length, and thus sends it to the peroxisome. So dicarboxylic acids, again, have a carboxyl group at both ends of the fatty acid. They are targeted to the peroxisome for beta oxidation. They do not actually form lipid droplets when we get them in our diet, and they're not packaged into lipoprotein particles. So they're really funky. They're more soluble than a regular fatty acid, so they travel more freely through the bloodstream, and they don't act as much like fat storage as a regular fatty acid would. There's also evidence in the literature to show that they may upregulate peroxisome function and biogenesis. And more importantly, they've been used in the past to treat animal models of obesity and type 2 diabetes. So they improve steatosis and glycogen storage and things like that. So these dicarboxylic acids may be a really beneficial easy way to upregulate peroxisome function and biogenesis, and thus avoid some of the peroxisomal beta oxidation process to ease the burden on the dysfunctional mitochondria of the RPE. These are all really preliminary thoughts that we're having, and we're currently investigating exactly how dicarboxylic acids are beta oxidized in the RPE specifically. So when we became interested in this, we looked into the literature. Sorry, we're going to redirect here. DCAs also have a lot of energy potential, even though they almost seem too good to be true. They travel through the bloodstream really easily, and they're much more soluble than other fatty acids. They do provide a lot of energy to cells, which we've seen in those animal model treatments of obesity and type 2 diabetes. When we run C18 dicarboxylic acid specifically on Recipher compared to palmitate, it rises more slowly at this early time point, but it just keeps increasing OCAR for a long time. It just goes up and up and up and up. And so this indicates that it does provide a lot of energy to the cells. So maybe in the future we can use these to improve peroxisomal beta oxidation and lipid metabolism in general in the RPE of AMD patients. When we looked into the literature into dicarboxylic acid beta oxidation, we found a bit of a murky story. The literature is not very conclusive as to how dicarboxylic acid beta oxidation ends. So some of the papers that we've read suggest that peroxisomes only produce adipic acid, which is a C6 dicarboxylic acid, from the beta oxidation of longer chain DCAs. And that's pretty common in the literature to say that peroxisomes produce adipic acid. However, short and medium chain dicarboxylic acids can actually enter the mitochondria and produce succinate, which is a 4-carbon dicarboxylic acid. But the answer to this question is definitely a little bit murky, and we're not entirely sure how it works, or if it's different in the RPE than it is in other tissues. It's certainly a different process than the beta oxidation of regular monocarboxylic acids. So I've been running a lot of different dicarboxylic acids in our Recipher device. And based on my results, what we're arguing now is that C6 dicarboxylic acid or adipic acid can actually be beta oxidized down to succinate in the peroxisome. And the reason we're concluding that is that when I ran C6 monocarboxylic regular fatty acid compared to the dicarboxylic acid, these results indicate to us that C6 monocarboxylic acid rises similar to palmitate and other long chains that we've seen, and later comes back down below the control. This is more of a muted rise in OCAR because there's just not as much carbon in C6 compared to palmitate. However, the dicarboxylic acid version of this fatty acid just keeps rising and stays at a higher OCAR than the control throughout the run, which indicates first that it's being beta oxidized most likely, but it also suggests to us that it's being beta oxidized in the peroxisome. So this is really niche, big news in a small field of research to suggest that peroxisomes beta oxidize dicarboxylic acids all the way down to succinate, which is a 4-carbon dicarboxylic acid that is also a TCA cycle intermediate. We're not entirely sure if it actually feeds the TCA cycle, but we're hesitantly concluding before we do acylcarnitine profiling or DCA profiling that peroxisomes break down dicarboxylic acids all the way to succinate rather than adipic acid. And again, that's very niche news in a small-ish field of research. So in conclusion, we're determining that peroxisomes in the RPE can beta oxidize long chain fatty acids. Using our Recipher, we've been able to, as far as we can tell, differentiate between mitochondrial and peroxisomal beta oxidation, while we get this more holistic view of how cells are utilizing the fatty acid and other substrates that we feed them. And our new evidence suggests that maybe adipic acid can be beta oxidized in peroxisomes, at least in the RPE. In the future, we'll be definitely doing more acylcarnitine profiling and other tagged fatty acid trace studies using long chain fatty acids and dicarboxylic acids. We've also been working on developing that negative control for peroxisomal beta oxidation based on an iPSC line that our research faculty member Jatao has been differentiating to RPE. And we're going to be investigating the effects of dicarboxylic acid supplemented media on peroxisome function and biogenesis in vitro with our RPE cells and some a liver cancer cell line as a physiologic control because peroxisomes are bigger in the liver. So we have a lot of really exciting rabbit holes that we've been going down within this project that I'm really excited to tackle this year and in the coming years. Huge thanks to my lab, especially John, our postdoc, who kind of got this project up and running before I took it from him so he could do some other stuff. And Jason, who's been a really wonderful mentor, as well as some of our collaborators, especially Matt and Constantine and the whole Benson lab. And big thanks to Dan, who not only hosts this seminar series, but also puts up with our acylcarnitine profiling requests and is a really great collaborator of ours. So thanks, everyone. I'm happy to take questions.
All right. Any questions? I know we had several that were asked during the talk as well.
Well, I got a technical question actually. So you guys showed with the Recipher that you actually get a pretty steep gradient of oxygen, and where the cells are, there's actually very little oxygen, which is probably not physiological. And I wonder if you did these experiments in a incubator that had like 95% oxygen instead of 21% oxygen, everything might just go faster, because maybe the rate limiting step for fatty acid oxidation is actually the availability of oxygen, because it's down to the bottom of the well and you're not stirring the well or anything like that. So I wonder if that would make your days shorter.
No, that's a great point. I don't think we've tried a hyperoxic environment. We certainly actually do a lot of hypoxic studies, not me personally, but some of our other lab members use hypoxic chambers, as well as a larger media volume that limits the rate of oxygen transfer through the media column. So we'd have to look into that, but that's a really great idea. Maybe it's just the availability of oxygen that's the rate limiting step.
Yeah, at the bottom of the well, they're probably not anywhere close to what they would be in an eyeball. So
True. Yeah, we do have, I use a pretty small volume of media. I typically use about 80 microliters in these 96 well plates. And we've shown that between about 55 and 85 microliters, we're not getting a large change in the transfer of oxygen from the top to the bottom in like control conditions. But I've found that going up to the top of that kind of gradient closer to 80 microliters makes it a little bit more consistent and limits evaporation effects. We do have water wells around those specifically aligned cell wells to inhibit evaporation effects, because that definitely can happen. So we've looked into it, and Jason, you can talk more about it if you want, but that's a great point.
It's just very briefly, Jim, of course, it's a very good point. We don't want to be in O2 limiting conditions. We also want to be at about physiologic pH or oxygen if we can. Fortunately, we've worked out the system well enough that the volume we put on, we can titrate the O2 availability down at the RPE. And this system allows us to calculate what the O2 is at the RPE, and at the concentrations we're at at the volumes we're at in the 55 to 85 microliter range, we are at physiologic O2 concentrations
Right,
at the level.
but that was without the palmitate there, because when you have the palmitate, the OCR goes up. And so
That is definitely a very good point. We can easily go back. It's a good point for us to go back and do the calculation, because that probe is in there. We know the O2 tension and we know the diffusion gradient. So you can just calculate the gradient all the way down to the bottom and calculate it. And that's a good point. We should do that to see how much the O2 at the cells has dropped when we put in the palmitate. That's a great point.
Yeah, based on the result, it's when you add when you added the palmitate, the oxygen added the cell surface is still enough. It's not a zero. Yeah.
But we should be precise and report back to you to know what it actually is. So thanks for that point, Jim.
Oh, any other questions?
Sorry, I know that we're well over time.
That's all good. I'm gonna let everyone go then. Thank you so much, Abby. This was a wonderful talk.
Yeah, thank you, everyone.